In this screencast, we're going to continue talking about the cell cycle, and specifically here we're going to talk about meiosis and the formation of sex cells in sexual reproduction. As a review of mitosis, remember that the result of mitosis is to produce identical daughter cells with the exact same information in each daughter cells. The daughter cells are clones or exact copies of one another, and they are exact copies of the parent cell. So there's a number of organisms, specifically yeast and many protists that are single-celled eukaryotes that use mitosis for asexual reproduction, but as well as some multicellular eukaryotes that are still simpler organisms. This budding that occurs in Hydra is an example of asexual reproduction. Should be able to discuss the disadvantages of asexual reproduction, but also the advantages of asexual reproduction. Of course, disadvantages would include that there's less variation produced, but advantages are, well, you don't have to go through the trouble of finding and fighting for and winning a mate. So a look at this same process of mitosis is, what about if that's the way that it worked with the rest of us? What if complex multicellular organisms want to reproduce? Well, we know that you need to join an egg and a sperm cell, and since cells are made by cell division, can we make egg cells and sperm cells by mitosis? And of course the answer is no. If we did, the egg cell that has 46 chromosomes for a human, and a sperm cell would have 46 chromosomes for a human, and then you'd have a zygote at fertilization that have 92 chromosomes. So the math, the chromosome math, wouldn't work. We haven't talked a lot about chromosome numbers yet and the amounts of genetic material, but we do know that human body cells have 23 pairs of chromosomes, and they're all the same. But it's important to know that we can't mess with the chromosome number and the genetic material. And I think of it kind of as a lot like music. So you might have heard Beethoven before, and Beethoven music is it's very distinct as being a classical music choice. You might be familiar with Beethoven's Fifth. Well, as opposed to that, also a brilliant musician would be Kurt Cobain, who had his own style that was pretty distinct. Either one of them are pretty recognizable. But if you mess with the chromosome number, sometimes the genes and the information get screwed up, and it would be kind of like trying to listen to both at the same time. That wouldn't work so hot. So humans always have, in normal cells, they're going to have 46 chromosomes, which occur in pairs. Here we're looking at a female karyotype, a look at all of the chromosomes. You got chromosome pair 1, 2, 3, 4, 5, 6, and essentially they're just lined up. The longest pair is called number 1, and the shortest of the body chromosomes, the somatic chromosomes, would be 22. And then here is circled, a female is going to have what are called 2 X chromosomes. Males who are going to have all of the same numbers of chromosomes, and you're going to have the same chromosomes as the females, except instead of having 2 X chromosomes, they have an X and a Y chromosome. So there's going to be a lot more on that later as we continue into studying genetics. All of the chromosomes that I just showed you in the karyotype, they occur in pairs. And those pairs are referred to as homologous. And that really means, homo or homo means same, and homologous basically means the same or similar but yet different. So they occur in pairs, and they carry the same matching genes. It's just that the genes on each of the chromosomes are not necessarily identical. So in order to actually make sperm cells and egg cells, or reproductive cells, or gametes as they're called, we actually have to reduce the number of chromosomes and cut it in half. That way, at fertilization, we will be back to the normal number of chromosomes, and we won't end up with information overload or genetic material overload, and we won't be ending up listening to Smells Like Teen Spirit and Beethoven's Fifth. So we focus here on the process of meiosis. Meiosis is it's a cell division. It's part of cell division that ends up producing gametes. So instead of getting a cut on your knee and making more skin cells that are identical to the old skin cells, in meiosis we produce gametes, which are actually unique. And so we go from a diploid to a haploid situation, or in other words, we can go from 2N to N. Specifically with humans, we go from 46 chromosomes, or 23 pair, to just 23 individual chromosomes. We half the amount. So meiosis actually reduces the number. At fertilization again, we're going to restore that diploid number, or we're going to restore the 2N situation, which in humans would be 46 chromosomes again. So take a look at this life cycle in organisms that reproduce sexually. You can stop and pause the video and just follow the cycle. A term I want you to be familiar with is reduction division. And what that means is that the results of meiosis, the cells that result from meiosis, there is a reduction in the genetic material. It is cut in half. So there's two divisions in meiosis. Meiosis and mitosis, they sound the same. There are similar stages because of how they evolved, but we need to keep the two straight, and they are equally important to life and to cells. So in a brief overview here, you're going to see some things that are pretty familiar to you from mitosis. Instead of just the IP mat here, though, we'll see that we kind of have a doubling or a repeat of the processes. So we've got interphase 1, prophase 1, metaphase, anaphase 1, but we also have a second division. So we end up with actually 4 daughter cells when we started with 1. That's different than mitosis. Remember, as we just said, there's going to be a reduction division in meiosis. So in order to do the math right, or to keep consistency or continuity with the DNA material, the genetic material, they still are going to reproduce. The cells are still going to reproduce or replicate the DNA. They're going to replicate the chromosomes. But because there's going to be 2 divisions and only 1 replication, you're going to end up with daughter cells that have half the genetic information. The first step of meiosis is, again, duplication of the DNA. And as we just said, because there's going to be 2 divisions this time, we've got to actually duplicate the DNA so that the accounting is correct with the resulting cells, the daughter cells of meiosis. The first division of meiosis is going to separate the homologous pairs. So when the DNA duplicates itself, you're actually going to have what are called a tetrad, where you're going to have homologous pairs of chromosomes. Remember, similar but not the same. The homologous chromosomes are going to be lined up next to each other eventually in metaphase. But the first step is to replicate the DNA. You double it. Then in meiosis 2, or the second division of meiosis, the sister chromatids are going to divide and separate from one another, and we'll end up with the 4 daughter cells. As we start to put this together, we'll see that there's meiosis 1 and there's meiosis 2. In meiosis 1, you're going to see the same phases as we did in mitosis. Interphase, prophase, metaphase, anaphase, telophase. But then there's going to be a second division in prophase 2, metaphase 2, anaphase 2, telophase 2. An extremely important part of meiosis is a concept called crossing over, and we'll look at this in more detail, of course. But in meiosis, when chromosomes are going to be next to each other, when they're going to have a tetrad, early in meiosis, in prophase 1, these may not all be lined up in the middle of the cell, but the homologous chromosome pairs are going to be next to each other. And during that point, there's going to be small spots where there's kind of an attachment or a contact point between the two homologous chromosomes. Those contact points are called chiasmata, and at that point is where some crossing over can take place. That crossing over is where the DNA, that part of the chromosome, that chunk of the chromosome, it breaks off, and they actually switch places here. So you can see in this model where specific parts of one chromosome break off and they swap spots. Now the implications of that is that the genes, they may be similar, but they might be different forms of the same gene. So much as you see in a model here, you see the different color in each of these homologous chromosomes, the overall gene makeup and gene sequence and DNA sequence has now just been shuffled. And without getting into a lot of detail of the steps of this, again, just by looking at the models here, where the arms of the chromosomes, of the homologous chromosomes, they make contact, and there can be a breaking off point. And what that does is forms new combinations of traits. And actually crossing over can be very advantageous. Sexual reproduction in a species benefits from genetic variability. So here you should pause the video and just take a look at the comparison between mitosis and meiosis. Many of the stages are the same, but the big difference is the resulting daughter cells and the differences between the daughter cells. In mitosis, we end up with identical daughter cells that were identical to the original. Of course, this already has shown replication. In meiosis, the daughter cells, there's 4 of them, and each of them is unique from each other and actually has half of the genetic material as the original parent cells. So this slide is just kind of a summary of the events of mitosis versus meiosis. Pause the video and review the differences. And of course, both processes are applicable or important to life. With meiosis, we have fertilization that produces a zygote, and then by mitosis, there's going to be growth and development through the lifetime of that organism. In this screencast, we're just going to take a brief look at the value of sexual reproduction, but these are really 3 very important points of understanding. In fact, these are addressed in the AP curriculum as being learning objectives, that the value of sexual reproduction really is all about producing genetic variation. Genetic variation is good for the health and the long livelihood of species. So genetic recombination occurs when we have independent assortment. In the phases of meiosis, we actually have random alignment of those homologous chromosomes, and so we don't know really which one is going to end up into which gamete. There's also going to be crossing over. That just further mixes the alleles, those are forms of genes, mixes up the genetic material, so it just shuffles the genes that end up in the gametes or the reproductive cells. And then there's simply random fertilization. And what we mean by that is that in organisms that reproduce sexually, we really don't know which male gamete is going to fertilize which egg cell. And so there is a whole shuffling and randomness to the biological explanation of sexual reproduction. So in humans, the independent assortment of chromosomes, meaning the shuffling and the division of chromosomes, the picking of teams as to which chromosomes are going to end up in which sex cell, it has everything to do with the way the chromosomes randomly line up in metaphase. And this is a huge number, over 8 million different combinations for human chromosomes, because there's 23 pair of them. There's over 8 million different combinations in gametes just from independent assortment alone. On top of just independent assortment, We also shuffle genes just because of the phenomenon of crossing over. So we actually change the chromosomes. And what's interesting about that is that we haven't studied this a lot, but if you change the chromosomes, you are obviously changing the genes and the DNA that make up those chromosomes. And back with humans, since there are two parents and they both have the likelihood of this huge number, over 8 million opportunities to produce a certain gamete, the essentially random fertilization of which sperm and which egg between a couple are going to fertilize, we've got a lot of combinations, a lot of diploid combinations of a fertilized cell, which becomes a zygote. Lastly, we have just these two slides again that discuss and summarize the meiosis phases. You're going to be quite familiar with this in the projects and the work that we do, but we want to be here with meiosis 1 and 2 and be familiar with the end products or the daughter cells and how they relate to the original cell.